CRISPR is the most promising gene editing method currently being researched by scientists to alter the genome. The term CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats and is associated with protein Cas9. This system is found naturally in bacteria and other prokaryotes and is involved in their immune defense. These prokaryotes use CRISPR to recognize the DNA of invading viruses and use Cas9, an RNA-guided DNA endonuclease enzyme, to cut up their DNA upon invasion in order to disable them. This natural mechanism has been utilized and modified by scientists to edit recessively and dominantly inherited monogenic disorders, such as Duchenne muscular dystrophy. Duchenne muscular dystrophy, which we will refer to as DMD, is a single gene disorder caused by different mutations on the DMD gene. This is the largest known gene in our genome with 2.2 million base pairs. These mutations inhibit dystrophin production. Dystrophin is a protein essential for muscle function, and without it, humans experience muscle degeneration, loss of mobility, and a premature death. Men are more susceptible to this disease, affecting approximately 1 in 3,500 males, as the gene is located on the X chromosome. DMD is a recessive gene mutation, so if males inherit the defective gene, their phenotype will show the physical traits of the disease. If a female inherits a single defective gene, their phenotype will not express DMD unless both X chromosomes hold the defective gene. There is currently no cure for this fatal disease, but CRISPR has potential to treat DMD as it can effectively cut out the defective exons, allowing dystrophin to be produced. This has proved to be successful in the MDX mouse model. The MDX mouse model of DMD has a nonsense mutation on exon 23. This mutation is when a change of a DNA base causes a premature stop codon in the DNA sequence, terminating the appropriate polypeptide and therefore correct protein production. If this exon is removed, then the protein can be translated and hence produces a functional protein, aka exon skipping. Currently, adeno-associated virus, AAV, is being used as a delivery mechanism of a CRISPR-Cas9 because it does not get rejected by the immune system. In this example, the Cas9 protein acts as a molecular scissor that cleaves the DNA at a specific target, which in this case is exon 23. A sequence of guide RNA, shortened to gRNA, with 20 base pairs forms a complex with Cas9. The gRNA base sequence is complementary to the sequence of DNA that is being removed, so it directs the Cas9 to this sequence. When the complex recognizes a short DNA sequence known as a protospacer adjacent motif, a PAM, next to the target sequence, it will bind to this. The gRNA then proceeds to unwind and separate the double helix by base pairing with the target sequence on one strand only. Once the double strands of the target sequence are separated into two single strands, the Cas9 cuts the target DNA sequence on both helices, producing a double-stranded break, DSB. The cell will want to repair the DSB. In the case of DMD, this will be by non-homologous end joining. The cell will either insert base pairs or delete base pairs surrounding the break in an attempt to rejoin the DNA strands where there is no repair template. The DNA strands are effectively rejoined without inclusion of exon 23. The base pair is naturally joined by a hydrogen bonding that is promoted in the cell environment, resulting in a double helix. The addition or deletion of nucleotides is not specific or controlled, therefore can cause mutation. When this occurs in non-coding introns, it does not impact protein production. When functional dystrophin is produced, muscle function can be improved. Although these mice models have different disease to human DMD, they show how mutations can be removed using CRISPR to form a functional protein from a gene and correct disease. Though this gene editing method has proven successful in MDX mice tests, in humans there are limitations to this technique. A problem arising is that Cas9 can sometimes cut DNA off target. Sometimes it can cut up to 150 wrong targets, which can lead to further unwanted mutations. It is unknown the long-term effects of these mutations. Organisms with larger genomes have a greater probability of similar DNA sequences to that of the target existing. This makes the chance of unexpected mutations and effects much larger in humans than in mice, which have a smaller genome size. An issue with AAV being used as a delivery mechanism is that there is a chance of a host immune response. Antibodies recognizing AAV are naturally found in many individuals, so rejection by the immune system can interfere with the therapeutic purpose of CRISPR. In conclusion, CRISPR is an exciting new gene technology that potentially can cure life-threatening diseases, but it still has a long way to go before that can be a reality. The successful demonstrations from mouse models, however, are promising for a future where this technology aims to be a common medical tool.